All right, good morning, students. As you see up on the screen, you should have turned in your homework to the basket at the front of the room. You need to make sure that you picked up your assigned sticker, so either an A, C, a T, or a G. Your groups are listed here, assigned which sticker you should have. And also you need to make sure that your science notebook is on your desk and you have something to write with. Okay, looks like everyone has what they should. Okay, so now look at this picture that's up on the screen and think about these few questions. How is the ladder put together? Are there any special elements that provide support? How are the rungs attached to the outside rails? And what concept in science do you think that this ladder could represent? Hopefully your answers are that the concept is similar to DNA. Hopefully that you see the rungs are attached to the outside rails. There are screws that provide support. That also is what holds the ladder together. Okay, so now we're going to do an activity. So in your lab groups, make sure your sticker that you picked up is placed on your right fist, on the back of your hand. Find an area in the room, and then we're going to go through this activity together. Okay, so repeat after me. So, students, you're gonna say, I am a nucleotide. I have a phosphate on my fist, so you're gonna stick out your left arm. So your phosphate is on your fist. Then you're gonna say, I have a sugar on my shoulder. And you're gonna take your right fist and hold it up to your shoulder. And then you're gonna say, and a nitrogenous base, and you're going to punch out your right arm. In your lab groups, you should be connected. Your fist should be pointing at another one of your lab partners. So now let's do it all together. Ready? I am a nitrogenous base. I have a phosphate on my fist, a sugar on my shoulder, and a nitrogenous base. I am a nucleotide. Very good. Return to your desk, and we're going to continue on with our lesson. Okay, so we just finished our I am a nucleotide activity. So for this portion of the lesson, for the dyslexic students, at this time I would hand them a copy of the notes that we're fixing to go through. And also for our ELL students, I provide visuals within the PowerPoint presentation to go along with the notes. Okay, so students, now we're going to talk about the details of what we just went through with our I am a nucleotide activity. The first thing we're going to talk about are the complementary base pairs. What are they and why are they important? So complementary base pairing is a phenomenon where adenine only pairs with thymine and cytosine only pairs with guanine. This is very important in forming that helical structure that we know DNA to have. And it's also important in DNA replication. Now, DNA replication is something that we're going to talk about in a later lesson, but it is very important to know that one of the reasons is through complementary base pairs. So next we have the purines. So this is one of the molecules that we have within DNA, and it's the larger 6-member ring that's fused to a 5-member ring. And the two examples that we have of the purines are going to be adenine and guanine, as you can see right here. Next are the pyrimidines. This is the other molecule that we have that makes up DNA. And these are smaller, they're only a 6-member ring, and they're made up of carbon and nitrogen. The pyrimidines are thymine, cytosine, and uracil. So uracil is found only in the RNA, but we're going to talk about that later on when we get to RNA. So for now, let's just focus on thymine and cytosine. Chemical bonds. We have chemical bonds in DNA, and these are very important. So this is what's going to hold the two strands of DNA together. And these bonds are found between the nitrogenous bases. So go back to your activity when you had your fists out pointed at your partner next to you. In between your two fists, you're going to find bonds. And those are going to be hydrogen bonds. And hydrogen bonds are very weak bonds, so they're easily broken, which again, that's going to play into when we talk about DNA replication. So in between adenine and thymine, you're going to find 2 hydrogen bonds. And then between cytosine and guanine, you're going to find 3. So next we have, how do we read a DNA strand? You know, you have all of these molecules in a line, but we don't know exactly how scientists read them. Well, we do because we know that they're going to read them from the 5' end to the 3' end. So essentially, DNA has built-in directionality, and that means that on one side of the strand, it's always going to be read from that 5' to 3'. But then if you look at the other side, that strand is going to be read from the bottom up. But as long as it's going from 5' to 3', we know it's going to be read in that direction. Also, at the 5' end of the DNA strand, you're going to find a phosphate group. And that's only going to be found on the 5' end. On the 3' end, you're going to find a hydroxyl group. And that's only where you're going to find the hydroxyl group, is on that 3' end. Okay, so now we're going to take what we just learned from our nucleotide activity as well as our lecture here, and we're going to take all of that information and apply it to our own model. Okay, so students, repeat after me. We just went through notes on DNA. Now we're going to build our own models, and these are related because it's taking information that we just learned and applying it to our model. Okay, students, so now we're going to build our own model of DNA. For this portion of the lesson, for the ADHD student, I'm going to provide step-by-step instructions through the making of their models. Okay, so in front of you, students, you should have 2 red pipe cleaners. You should have had 1 orange, 1 blue, 1 yellow, 1 green, and 1 black pipe cleaner. However, we cut the pipe cleaners into 4s and then 8s. And then you should have had 4 white pipe cleaners that we cut into 4s as well. I'm wearing safety goggles because since we're working with scissors, we have to make sure that we're taking all necessary precautions to protect ourselves. So especially our eyes, since they are sharp objects. Okay, so let's get started. So you're going to take your 2 red pipe cleaners and put them down. So from the lecture, we knew that adenine pairs with thymine and cytosine pairs with guanine. So to start building our model, I'm going to take one of my adenines and I'm going to wrap it around my red one. Okay? So then I'm going to take my thymine and I'm going to wrap it around the other one. So remember when we talked about our purines and our pyrimidines, we knew that the purines were larger than the pyrimidines. So since adenine is one of our purines, we have to make sure that that one's longer. So that's why I'm wrapping the thymine here shorter than I did the adenine. So when you're done wrapping them, you should have 1 longer one for the adenine and 1 shorter one for the thymine. Now, what did we say went between the 2 nitrogenous bases? That's correct, a hydrogen bond. So we're going to take our black pipe cleaner and we're going to wrap it around the 2 nitrogenous bases. Now we're going to wrap it thinly because remember, like we said in the lecture, hydrogen bonds are weak bonds. So we're going to wrap it just to hold those 2 together. Okay, so you should now look like this. Okay, and then think about our nucleotide activity. What did we have on our shoulders? That's correct, we had a sugar. So you're going to take your white pipe cleaner that represents our deoxyribose sugar, and you're going to wrap it around the rung. So you're going to have the white sugar wrapped around. Then you're going to have your nitrogenous base, your hydrogen bond, your other nitrogenous base, and then another deoxyribose sugar, because what's on one end or one side of the strand is going to be the same on the other. Okay, so then you can take your cytosine, which is the yellow one, and you're going to wrap it around the row. Remember, cytosine is a pyrimidine, so it's going to be shorter. And then what pairs with cytosine? Guanine, that's correct. And guanine is our purine, so it's going to have to be longer. Okay, and then what do we need to add? A hydrogen bond, correct. And that will hold these two together. And it's thin because it's weak. It's a weak bond. It can be easily broken, which is very important when we start discussing DNA replication. And don't forget to add your sugars to the outsides. And our sugars are the deoxyribose sugars. When we talk about RNA, you're going to have a different type of sugar. So far your model should look like this. Okay, so our completed model should look like this. Remember, you have your phosphate backbone, you have your deoxyribose sugars, you have your two nitrogenous bases. The black indicates the hydrogen bond. And then you have the longer ones representing the purines, the shorter ones are the pyrimidines. And we know our purines are going to be adenine and guanine, and our pyrimidines are thymine and cytosine. Okay, so now the other part that we talked about in the lecture was our 5' and our 3' end. So if up here is our 5' end, this end is going to have the phosphate group attached to it. So we know down here is going to be our 3' end. So take your scissors, go to your 3' end, and you're going to chop it off short. So we know that this little extra piece up here is our phosphate group. Now on the opposite side, down here is going to be your phosphate group. So this is now your 5' end, and this is your 3' end. So again, take your scissors, chop that top part off. Okay? So now I have a 5', 3', 5', 3'. So now the other thing about DNA is it's not shaped straight like this. It's actually twisted. So where you can take your model and you can twist it to kind of make that helical structure that we mentioned, because DNA is a double helix. So see how it has that helical structure? So now our model of DNA is complete. So once you have this completed, there's a little folder in your science notebooks. You can untwist it so that it's flat and then place it in that folder in your science notebook. Okay? Okay, so at this point, all of our DNA models should be in the science notebooks put away. As the teacher, at this point in time for the lesson, I would walk around and hand out the blank diagrams of DNA that the students are going to be evaluated on. They should have no notes on their desks, they shouldn't have their models out, but yet they should be able to fill out their blank diagrams with 80% accuracy at least. For the ELL student, a word bank is provided, and also for the ADHD student, this assignment is tailored to them. So it's going to be printed in color, it's going to be engaging where they're not losing focus and they can they can provide their answers for the assignment efficiently. Okay, this time everyone should have turned in their assignment to the basket. I'll have those graded and back to you by the start of class tomorrow. So now I'm going to ask you a few questions. We're going to use the popsicle method. So I would randomly pull a popsicle stick out of a cup. So student 1, I would ask them, what did you learn today? Student 1 would hopefully respond that they learned all about the structure and the components of DNA, because that was our teeth that we were focusing on. So then I would pull another popsicle out of a cup and I would say, student 2, what was the purpose of the lesson today? And they would say to learn all of the basic, functions and structure of DNA with their components, and use that as a fundamental building block so that when we start talking about more complex aspects of DNA, they would be able to understand, why complementary base pairs are important, why we have weak hydrogen bonds, etc. And then also, this would be a chance for students to ask me any questions. So I would ask them, is there any clarification that you need from this lesson today? And at this point, students would raise their hand. I could call on them. They would ask me questions and I'd be able to provide feedback. We'd be able to talk about what we learned. I could refer back to the model. We could refer back to the nucleotide activity. All of these aspects of the lesson today we would be able to use to answer students' questions.